Salam sejahtera dan selamat para penonton webinar Program Magic Revision Siri 2 slot keempat SB015 Biologi di YouTube live stream. Saya, Madam Hwee Cheng dari College Matrikulasi Johor selaku moderator malam ini mengalu-alukan pelajar-pelajar dari seluruh College Matrikulasi Kementerian Pendidikan Malaysia untuk menyertai program kami pada malam ini. Untuk makluman para penonton, program ini merupakan anjuran bersama pensyarah cemerlang dari College Matrikulasi Selangor dan College Matrikulasi Johor. Pasukan kami terdiri daripada 6 orang pensyarah cemerlang dalam bidang kepakaran matematik, kimia, biologi dan fisik. Dalam setiap slot, kami akan berkongsikan tips penting menjawab soalan PSPM mengikut pengalaman mengajar dan bidang kepakaran kami masing-masing. Tujuan utama program ini dilaksanakan adalah untuk membimbing para pelajar Kolej Matrikulasi KPM membuat persediaan rapi dalam menghadapi peperiksaan semester program Matrikulasi 1 yang akan berlangsung mulai 6 Januari 2021. Ya, pelajar-pelajar sekalian, adakah anda bersedia untuk menghadapi PSPM 1? Kalau yang bersedia, boleh tak para pelajar dekat YouTube Live type respon sikit? Kalau yang sudah bersedia, boleh tak you type 1? Kalau yang masih belum bersedia lagi, you typekan 0. Okey, kita nak tengok berapa orang pelajar telah bersedia. Ada tak respon kat sini? Ada. Okey, so webinar malam ini terbahagi kepada 2 slot. Slot pertama adalah sesi pembentangan oleh penceramah, manakala sesi kedua adalah sesi soal jawab. Semasa sesi pembentangan, pelajar hanya dinasihatkan untuk mendengar penyampaian penceramah dan tiada sebarang interaksi antara penceramah dengan pelajar. Meminta kerjasama daripada semua pelajar untuk menempuhkan semua perhatian semasa sesi perkongsian berlaku. Jika ada sebarang soalan, bolehlah menulis soalan anda di live chat YouTube. Jika masa mengizinkan, penceramah akan menjawab semua soalan semasa sesi soal jawab nanti. Okey, yang ini merupakan Madam Nashimah Habeeb dari College Matrikulasi Johor. Okey, say hi kepada Madam. Boleh? Yang kat luar? Hai. Okey. Beliau merupakan pensyarah yang berpengalaman banyak dalam subjek biologi dan untuk pengetahuan anda, beliau mempunyai 16 tahun pengalaman mengajar subjek biologi dan 10 tahun pengalaman mengajar di Kolej Matrikulasi Selangor. Adakah pelajar-pelajar di saluran live YouTube ingin menerima input daripada Madam Nashimah? Okey, kalau anda sudah bersedia, boleh tak anda type 1, respon 1. Okey, kita Madam Nashimah, okey, kita menunggulah sekurang-kurangnya ada 50 pelajar respon type 1 baru kita mula, boleh? Boleh. Okey, Madam Nashimah, sementara menunggu pelajar kita respon, saya ada satu persoalanan. Okey, antara banyak topik biologi ini, kenapa Madam memilih topik how to tackle biology PSPM questions based on DNA, recombinants and reproductions?
Okey. So kalau kita lihat dalam biologi sebenarnya ada 10 chapter. Okey, dalam banyak-banyak 9 chapter saja yang akan disoal, dan dalam 9 chapter ni 2 chapter iaitu DNA recombinant dengan reproduction membawa markah yang paling banyak. Okey, so kita memang nak skor bagi pelajar, peluang skor yang paling maksimum lah. Dan sebenarnya 2 chapter ni adalah mudah untuk dapat markah kalau betul-betul jawab ikut teknik. That's why saya pilih chapter ni.
Okay, so bagi saya sebagai pencara matematik, sejak kecil lagi saya berasa subjek biologi itu sangat susah. So saya harap melalui perkongsian Madam Nashimah ini, para pelajar boleh mendapat banyak ilmu, terutamanya tip-tip yang berguna untuk menjawab PSPM nanti. Okay, saya rasa pelajar-pelajar di ruang YouTube mestilah tak sabar dan dapat perkongsian ilmu dari Nashimah. So tanpa melengahkan masa, so saya menyerahkan masa ini kepada Madam Nashimah untuk teruskan perkongsian. Okay.
Okay, terima kasih Madam Lee. Okay, Assalamualaikum and good evening to everyone. Okay, so we meet up once again today, and today we are going to focus how to tackle the biology PSPM question. And actually I already shared the clone question to our Telegram, so I hope all of you already download and you try do yourself. And today we are going to discuss a little bit how to tackle the biology question, especially for chapter DNA and for the reproduction. Okay, so before we start, okay, allow me to just have a look. Okay, so what is the recombinant DNA technology? Okay, which is we already discussed in chapter 8 previously. Okay, so for this chapter, we are highlight more on 8.2 and 8.3, so which is the C3 and C4 LO. Alright, so method in the gene cloning and the method in the gene cloning, we also highlight about the tools. Okay, and then another the most important part is the application. Okay, so application, then we are going to discuss a little bit after this. Alright, so for the tools, okay, I'm just going to introduce roughly only. Alright, so tools here is the tools that need for the gene cloning purpose. Okay, so I'm sure you already understand about the tools, everything here. Okay, so we have the DNA cloning vector. Okay, so what is the important thing in the DNA cloning vector? Okay, so we have to understand why we are using the plasmid widely. Okay, so if you recall back your lecture note, everything, we have a various types of the cloning vector, but we choose plasmid, and most of the plasmid we extract from the E. coli. Okay, so you have to understand why the plasmid become the our main vector here. Okay, all right, and then the second one is the DNA target. Okay, so what mean by DNA target here? All right, so target here is a gene of interest. Okay, and sometimes you also can mention as a gene donor, DNA donor. Okay, so we can use a few terms here. All right, then we have a restriction enzyme. Okay, so the enzyme which we extract from the E. coli. Okay, so the E. coli. So we have a various types of restriction enzyme, but we only cover about EcoR1 and SMER1. Okay, why these two only? Because we want to differentiate their ends. Okay, their end here. All right, then we have a modifying enzyme. Okay, so modifying enzyme here we have a DNA ligase. Okay, and then we have another one is a Taq polymerase. Okay, so another one is the Taq polymerase. Okay, so the last one is the host cell. Okay, so the last one is the host cell. Okay, all right. So this is the steps in gene cloning. So like just now, okay, so we discussed about the what do you call it, the tools. Okay, tools need something like, let's say you want to make a baking. Okay, so tools is like the ingredient for your baking. All right, so step in gene cloning is how you are going to carry out the gene cloning. So altogether we have isolation of the gene, cut, cleave, insertion, transformation, and screening and amplification. So this one I think no problem with you. Okay, so we're just going to see another one. Okay, so this one is the production of insulin. So this one is under application. Okay, so this one is under application. Okay, so how from the DNA we are going to change into the mRNA, and from mRNA why we change back into the DNA, but we call as a cDNA or complementary DNA. Okay, so we are going to discuss this one in question. Okay, all right. Okay, so for the, let's discuss the first one. Okay, the question, okay, in biology, okay, before you discuss the question, okay, you have to focus, okay, focus. And I call the technique in answering biology as a IAIA method. Okay, what is the IAIA method? First, once you get your question, okay, once you get the question, okay, you have to focus, okay, identify all information given here. Okay, so identify all information given. Okay, for example, in the first question I'm sharing here is the, what do you call it, is the DNA cloning. Okay, so please identify, okay, what is the diagram is about, okay, what is the giving, the foreign DNA and so on here, right? So analyze the question, okay, so analyze the question. So the question is mentioning about, they are giving a DNA cloning. So the cloning here is the keyword already, okay? Then you have to record the information. Okay, what mean by record information here? Okay, so here you have to jot down. Okay, so once you get your exam paper, you just can contain. It's no problem. Okay, just contain with a pencil. All right, so you just stick first. Okay, what is the plasmid? Basically, what is the characteristic of the plasmid? Okay, everything. Right. Then this one is the most important part. Okay, so this is we call as a what are question asking about. Okay, so most of the students, they are very confident when answering the question, and but once they get the result, it's not so good. Why? Because normally they do not answer what is question asking about. They just answer what they know. Okay, so please be careful because biology is mostly theory. Okay, so once you understand the theory, how they twist the question is no problem. Okay, actually you just go back to the keyword. Okay, that's the important one. Okay, so maybe you can refer to your clone question. Okay, so I'm sure all the clone question is with you now. All right, so let's discuss a few questions here. Okay, so please focus on question number 7. Okay, so question number 7 in our clone is a DNA recombinant question. Okay, all right, so first look at here. Okay, so this question, okay, so show the parts of process involved in DNA cloning. Okay, so that is our question. All right, so now let's write the few important points here. Okay, so what is given here? Give it here. So they give here is a plasmid, and this one is the foreign DNA, or also known as the gene of interest. All right. So now they already give here eco R1. Okay, so you can understand eco R1 means what? Is our restriction enzyme. Okay, so it's our restriction enzyme. Okay, done. All right, then. Okay, so if you look at here, they are using the same restriction enzyme to cut both. Both means to cut the DNA and the plasmid. Okay, done. Then after that, you get the sticky end. All right. So this one is also giving you like a hint. Okay, so what the question will be about after this. And then after you do some process, okay, you know, once we mix between the plasmid and the gene of interest, so definitely you will get the here is the rDNA. Okay, and here what is the process? Insertion. Okay, So the first process is what? Okay, so it's a isolation. See, right? And then the second one is cut or cleave. So actually, we are discussing two things here. One is the tools, and the second one is the method. They cover together here. Okay, so the process here. Okay, then see, this is the bacteria cell. So normally the bacteria cell will be represented as our host cell. Okay, so this is our host cell. So how we introduce the bacteria to the host cell? So through the process we call as transformation. Okay, so you just jot down first. Whatever information you just cross in your mind, just jot down. Okay, so then we do the cloning. So then we do the process C. Okay, if you look at here, the process C, the bacteria containing the DNA recombinant grow. Okay, so they are select the DNA here. So this one will be the screening. See, right? Then after that, you do the purification or we do the amplification process. So this one will be the amplification setup. All right, so now you're already confident with your diagram. So let's see what the question is about. Okay, so state the characteristic of plasmid. Okay, once you get the question like this, okay, what is the keyword here? Okay, they give here plasmid. Okay, so plasmid, and they're asking the characteristic of plasmid. Okay, that is our keyword. Okay. So when we are discussing here the characteristic of plasmid, so who is our plasmid here? So this one is our, anyone? Yes, this is our vector. Okay, so you just can list down what is the characteristic of our vector here. Okay, so this is the question. All right, so if you recall back, okay, just erase everything. If you recall back how your plasmid look like, okay, so another thing in bio, you have to imagine. Imagination is really need for biology because you need to remember a lot of things here. Okay, so once you imagine, okay, it's like what? For me, okay, for me, I always remember the plasmid, okay, as lollipop. Okay, see, it's a colorful. So you can imagine, oh, it's a lollipop here. All right. So what is the content in our lollipop? All right. So see, look at the first point here. Okay, so this is my first point. Process of the multiple cloning site. Where is it? Okay, so this is the area multiple cloning site is located. Okay, so as you know here, what is a multiple cloning site? The site, okay, where they're able to receive the foreign DNA. Who is our foreign DNA? All the target gene, okay, the target gene or the donor, okay, or DNA donor. All is come here. Okay, so everything is, if a Hindi one like SPH, okay, then we have a EcoR1, everything. All is showing the various types of restriction enzyme. Okay, so the end means the cutting there. Okay, they can insert in this multiple cloning site. Okay, so this is the first characteristic of my plasmid. Okay, the second one. Okay, so this is the second point. Contain with origin of replication. Okay, so what is the function of your origin replication? Ori, or we call it. Okay, okay. Please say if you want to write in your exam as a ori, make sure you italic. Okay. Once you are writing, you cannot do italic, isn't it? So you just underline, showing CG. Okay. Ori means origin of replication means they able to initiate the replication starting from here. Okay, so that's another thing. And the last one, okay, they are consist of antibiotic resistant gene or also known as selectable marker. Okay, so where is my ampicillin resistant? This is the symbol we use. We call as a AMPR. Okay, so here also you can write as a, but make sure you write as a correct wording, AMPR. AMP small letter, R will be at the capital letter. Okay, so this one you need during your screening. Okay, during your screening. Okay, done. All right. So now you can ask me, Madam, why only 3 points? or how I'm going to make sure how many points should I put here. Okay, so if you look at these types of question, this is we call as an open question. Open question means they didn't give 2 or give 3. Okay, so they didn't give the number. Okay, so here you have to highlight how many marks they are allocated here. So 3. Okay, so this type of question is quite safe to you because you can answer more than 3. Okay, so if the question gives you already, okay, make sure you state 3 characteristics. If they already mentioned 3 characteristics, so you have to give 3 characteristics, or else there is a possibility wrong cancel right means once you give the 4th one wrong, so they will take another mark there. Okay, so just be careful with that types of question. But if the open question means, please look at what is the allocation marks given here. Okay, so based on that mark, you can identify how many points that you should in here. Okay, so this is one tips that you can score maximum mark for each question. Right? Alright, so now we are still using the first diagram. Okay, so we are going to discuss the second question. Okay, why the restriction enzyme EcoR1 is used to cut both plasmid and foreign DNA. Okay, the mark given is only 1. Alright, so if you look at here, alright, so they are using the EcoR1 here and also here. Okay, why they have to cut both? Okay, let's say you blur already, don't sweat. Okay, don't get nervous, just relax. Look at the diagram given. Everything is given in your diagram. It's a hint already. Look at here. What do you get here? Okay, so you can see, and they already give you the point. It's a sticky end there. Okay, so you just write down. Okay, so let's give another point here. To produce complementary, okay, sorry, this one, or compatible end. Okay, what mean by complementary here? Okay, so if you look at here, okay, let's say the orange color is your target gene. Okay, and let's say this one is our plasmid. Okay, so complementary means they have to be complement to each other. So look at here, they are complement to each other. So this is the complementary, or you can also use as a compatible. Right? So see it. Another thing is important during answering the biology is the terms. Okay, maybe you are confused. You say, okay, produce the same sticky end. All right, so please be careful. Kalau same sticky end, okay, so the same here is what? This is the same sticky end with this one. Okay, so you're having a problem. Okay, they cannot mix to each other. Okay, so please be careful. All right, so let's go to the next question. All right, just now we are discussing about the formation of sticky end. Okay, you understand already. Now, they ask you to write down. Okay, so how you are going to write down the cleavage product. So cleavage means here is a cutting process. Okay, we are discussing about the cutting process here. Okay, so using the EcoR1 in the space below. Okay, so normally they will give the space in your exam paper. So first, how you are going to cut down. Okay, the first thing you have to identify what is our restriction site for our EcoR1. Okay, so they just simply give you here EcoR1. So now you have to recall back. So for the EcoR1, the restriction, the palindromic sequence we call as GAATTC. Okay, and why I call it as a palindromic sequence? Because they will be read the same at both ends. Okay, so if you want to put the direction, it's up to you. Okay, 3 prime, so this one 3 prime to 5 prime. Okay, so this one is your palindromic sequence. Where is your restriction site? Site means where we are going to start doing the cutting process. So will be middle of the G and A and between with the G and A. Alright, so you just using your pencil or your pen, you just write draw the box like this. Okay. So you will get it's like a 2 box here. Alright, so 2 box here. Okay, so they say write down the cleavage product. What mean by product here? Is your fragment. Okay, if you show like this, it's still like the same box like that. So it's not the fragment. So to show the fragment, you have to separate. Okay, C, T, T, A. You just copy back. Okay, just copy back and do a box like this. Okay, and this one, okay, give a distance because we want to show there is a separation between these two fragments. So A, T, T, C, and here is the G. Okay, all right. Okay. Okay, I'm sure in the exam your drawing will be much more better than mine, eh? Okay. Okay, so can you see the clearing? Okay, normally here, I'm just giving you 1 mark, but actually you can go up to 2 marks here. Why I say you can go up to 2 marks here? First mark they're giving you, are you able to identify the palindromic sequence? First, are you able to identify the restriction site? Yes. Okay, so this one you're able to get 1 mark already here. Okay, and another mark they're giving you for the second drawing here. Okay, so you show there is a formation of 2 different fragments. Okay, so actually in this question you can score up to 2 marks. Okay, so this one is my the answer scheme. Okay, so see they give you the correct drawing, okay, the palindromic sequence with the GAA Then after you do the cut, so look at here. Okay, they give you the fragment. Okay, so you can draw like the table like this. Okay. Okay, then we go for the next question. Give the reasons why bacterial cell is suitable as a host cell. Okay, it's like a very simple question. Host cell. Okay, where is your host cell? Here. Okay, so now if you look at here, okay, they already give you the arrow, put it inside here. Okay, so I'm imaging. All right, so if I'm the host cell, okay, now I want to become a host cell. So what I should do? Okay, and if you look at this cartoon here, okay, they can appear the DNA inside their body. Okay, so the first thing is you must able to receive, okay, receive the DNA recombinant, okay, through the transformation process. Okay, I hope you understand what I mean by DNA recombinant here. Is the mixture between our target gene, okay, and our plasmid. Okay, alright, so it's mixed already. Okay, after you receive, okay, what you do, you able to maintain the structure of the DNA recombinant from one generation to another generation. Okay, so first receive, maintain, and the third one, you able to amplify the gene product from the DNA. Okay, so I give you the tips here. If host cell, make sure you remember these 3 RNA. Receive, maintain, and amplify. Okay, so you are able to understand that. I must receive first. Okay, so let's say the rDNA is your gas. I receive my gas. I maintain my gas. Then allow them to amplify in your house. Okay, you just can do any analog to make you fun and make you easy to remember. Okay, all right. Okay, is it okay, everyone? So far? Alright, so now let's go to second question number 7B. Okay, so 7B. So we show the step in gene cloning. Okay, this one is another types of question. Okay, so for bio, you can predict there will be a lot of diagram, pictures, or there can be a graph. Okay, so you just focus. What is the first things is given here? The given here is the gene technology. Alright. So identify the question. So gene technology here is related to application. Application form. Application. So you can expect the question here asking you about the cDNA, or they are asking you about the insulin, or they also can simply give any like a degrading bacteria or transgenic salmon, anything. Okay, so that means by gene technology here is an application question. Okay, so for first thing, analyze first. Right. So here is a gene to copy. All right. So this is from our DNA. Right. Then from DNA, okay. Now we get the single strand only. Okay. You don't know what strand is this. Okay. So then isolate the cell. So now degraded strand. And now they produce one strand, and this one is degraded. And from this one strand, now they become as a double strand back. So they become as a DNA back. Okay? And there is a process X, so suddenly we get many copies here. Okay. Alright? So, if you recall back, from DNA, which is the double strand, okay, now you're only able to get single strand. Okay, so this one is represent mRNA. Okay, how you know? This is the mRNA because of the strand. Okay, from antiparallel, there's double strand. Okay, now you get one. Okay, now from here, all right, so this is degraded here. So something is added here, so it's a degrading enzyme here. Never mind, they didn't ask. Just put to make sure you understand what the diagram is about. Okay, so the degrading enzyme is here. Okay, then here is the DNA. All right. So nampak is come back to here. So DNA, but I cannot call this one as a DNA alone. I must put here as a cDNA. C represent complementary. Why is it complementary? It's a complementary to my original DNA. Why is only complementary? Because during mRNA, we already cut intron. Okay, so it's present of the exon of the number. Okay, that's the things. Okay, then, so now we add some enzyme here. So then I get DNA, sorry, there must be a cDNA tadi. Okay, so what enzyme to adding another DNA strand? So, DNA polymerase here. Okay, and process X. Okay, so this one can be amplification. Okay, and there is another one terms is known as a PCR. Okay, but this one is not in our syllabus anymore. So we just assume this one is an amplification. So we make a lot of copy here. Okay, clear for this question. Okay, now let's look what is the questions about. Okay, explain what happened during B to C. So what's happening from this part until this part? Okay, so you go back here. Okay, so this is the information that you are going to use when you are answering the question. Okay, so given mark here 2. So you can expect there will be 2 to 3 points. Okay, so first, what is your first point? Okay, in B, right? So you just can use a very simple English, no problem. Okay. In B, single strand of cDNA is synthesized from mRNA. How you know? From my previous slide. Look at here. mRNA. Okay, so now this is my mRNA, and this one is the single strand of DNA. And another thing, please, you must highlight the word strand, single strand. Why you must highlight the word single strand here? Because we have another one in C, there is another second strand. Okay, you must highlight. In B, single strand of cDNA is synthesized from. Okay, you must mention who is the origin here. Okay, and from mRNA, the mRNA is degraded. So you just take the point here and you just add here. It's your point. Degraded by using degrading enzyme. Okay, let's say you forget your enzyme. Okay, you just can put as a degraded by the enzyme. Okay, 1 point is here. Okay, the second point now from here. So this one I'm talking about the B stage only. Okay, B stage only. Now this point I'm talking about the C stage. Okay, what's the C stage here? All right, so I already get the cDNA, but my cDNA is a double strand already. So reverse transcriptase is catalyzed the formation of second strand. Okay, so not the reverse transcriptase. All right, okay. So are you realize? I just simply put, okay, because you will be confused with the enzyme's name. I just simply put reverse transcriptase, but you have to understand when we are using the reverse transcriptase here. Okay, so what is the correct enzyme? See, DNA polymerase. That's why that is important to jot down first what is the information. So it's a reverse transcriptase. Okay, reverse transcriptase. Okay, so far okay or not? All right. So they catalyze the formation of second strand of cDNA from the single strand of cDNA. So you must make sure you mention from what to what, and who become as a template. Template here means they copy from single strand of DNA. Okay, all right. So please, please. All right. So this is the technique how you're going to score the value. Okay. Now let's go for the second question. Name the enzyme used to join DNA fragment. Okay. So join the DNA fragment. So this one is not related to our diagram anymore. Okay. So we can go back here, right, and state our hit word. Okay. Why it's not related to our? Okay. This diagram, okay, they are just isolating the target gene only. Okay, so you have to expect more from the question beyond the diagrams given. So if they give you here is only the target gene, so then you have to recall back. After we get the target gene, then we have to do all the process like isolation, then you have to do the cut, you have to do the insertion, then transformation and screening. Okay, so all that one you need to do. Okay, that's why we have to expect more. Okay, you can say, oh, it's not in the not in your diagram, but you have to prepare. Okay, see. So who will join your DNA? So this is the first point. You look at the point given here, 2 marks. Okay, so it's a DNA ligase. All right, and for second point, how the DNA ligase work? Okay, by join the DNA fragment. So this DNA fragment is, for example, from the gene donor and from the plasmid. Actually, you don't need to mention. You just make sure you just mention as a DNA fragment is enough. Join the DNA fragment by forming phosphodiester bond. So you get your second mark. Okay, so you already get your second mark. Okay, this one is out of the question. I just want to give you another input here. All right. Other than DNA ligase, we have another one we call as Taq polymerase. Okay, you remember this one, Taq polymerase? You have to isolate the Taq word. Why? Because this Taq polymerase is extracted from the bacteria from the hot spring. Okay, so they are doing the same work like a DNA ligase, but the difference is they can withstand with a very high temperature. Okay, but this one also the function during PCR. Okay, that's why this one is out of the question, just as your extra information. Okay, so now let's go. Okay, see if you look at here, the mark is increasing. Okay, so 1, 2, 3, and now we already go to the 4 marks here. Okay, describe how recombinant plasmid are formed. Okay, so please clear your mind. Your keyword here, recombinant plasmid. All right, what you can understand by the recombinant plasmid means is the combination between your plasmid and gene of interest. Okay, between these two, plasmid and gene of interest. All right. Okay, so now you have the problem where you are going to start your point. Okay, so am I going to start from insertion, or how the insertion I'm going to do if I didn't get my plasmid and gene? Okay, means I didn't get the fragment. Okay, so see, this is the fragment. Okay, so you have to what do you call it? You have to plan. Okay, you have to plan your answer before you simply write down. Okay, so avoid to use a lot of the correction tape, then you cut, avoid all that. Okay, because when there is too much of the writing there, maybe there will be a miss point there. Okay, so first you plan. So here is no diagram, isn't it? So no diagram, but how you are going to plan? You just plan at any corner of your exam paper. Just chonting, see now. Okay, so before I get my DNA recombinant plasmid, okay, what should I do? Okay, first I must isolate. Okay, if I didn't isolate my target gene and my plasmid, how come I'm going to get the rDNA? All right, isn't it? So isolate, and you have to understand. Isolation means what? I have to isolate 2 genes here. Okay, so one is the donor, another one is my plasmid. I must isolate both. Okay, Then after I isolate, so if you look at here, okay, this one is the reference diagram that I give you, but in exam maybe there is no diagram. Okay, so how I get this? So I have to cut, okay, using the restriction enzyme. Okay, restriction enzyme. Done. Two. Is it enough? Not yet. So now I want to get rDNA like this, one circle. Okay, so rDNA. What is rDNA? Don't get confused. The recombinant plasmid or nama lain is rDNA. Okay, so you can say any of the terms. Alright. And the third one, okay, so if you look at here, I should get like this, so I have to do the insertion. See, why you have to plan? Why you have to do plan? Because you can write down your answer according to the sequence. Why? If let's say you didn't plan, you're very excited writing about cut the DNA using the equal R1 then suddenly oh my god, after that suddenly you go to the insertion. Okay, insertion using the DNA ligase, then suddenly pop up another new idea coming. You say isolation. Then you simply the third point you write about the isolation. The problem is there because when they ask how to form the recombinant plasmid, means you have to give the correct sequence. See, the correct sequence. So by planning, you can get the correct sequence. Okay, so this one just planning. Maybe while you are writing your planning, suddenly you get an idea. So you just can erase and you do the new one, no problem. Okay, so please plan your answer. Okay, so this is my answer. So I can play first. Okay. So isolate the donor DNA and plasmid. See, my first point just now. I just jonty like that. So then you rewrite in the correct order. Okay. Then cut the donor DNA or gene target and plasmid. See, if you look at here, I underline the word and. Okay, it's not mean that N word is important. The important part is you must put both DNA and plasmid using the same restriction enzyme. Why? So to get the complementary, see, to complementary N. Okay, see, to produce compatible, or you can write as a complementary N. Okay, so 3 points already here. Okay, now the fourth point. Insert the DNA fragment into the plasmid and mix both fragments by using the DNA ligase. See, you see the clear the sequence of the answer. Okay, so now you already produce this one only lah. Okay, already produce your rDNA. Settle. Okay, all right. So this one is another tips. If you get the question without the reference diagram, okay, so please plan what you should include in your answer, but please don't take too much time spending to do your planning, please. Okay, just write the keyword, so then once you are transfer your keyword here, so you can write properly, okay, using the correct words. Okay, all right, so now it's keep increasing the mark here. Okay. All right, so 5 mark. Okay, explain the application of blue-white screening. Okay. Okay, blue-white screening in selecting. Okay, they already give you to select the recombinants. All right, so this one just as your guideline. Okay, so you know before we do the blue-white screening, what first thing you have to do? If you look at here, there is a transformation. Okay, so then for the screening, you have to transform the bacteria is plated on the medium with antibiotic and extract. So that diagram, okay, so this diagram. Okay, then first point here. Second point, all right, so you can get two screening. One is the blue color and another one is the white color. Okay, so for the blue color, so blue colony are formed. All right, so please do not confuse with the white color. Okay, please. That's why you have to do your planning first. Okay, just jot down, just something little bit. What is the point? Blue colony means what? The blue colony, the bacteria with non-recombinant plasmid have functional lexi. Okay, so the functional lexi able to produce beta-galactosidase, which hydrolyze the X-gal and form as a blue colony. That's the point. So you already get 3 points here. Okay, now let's look for the white colony. Okay, look at here, the white colony present. Bacteria with the recombinant plasmid have non-functional lexi, so they will unable to produce the beta-galactosidase, so cannot hydrolyze the X-gal, so they will be remain as white colony. Okay, all right. So if you confuse with the white and blue, just remember the X-gal is a lactose. Okay, so milk. You just imagine the milk is white color. Okay, so milk cannot be hydrolyzed, they remain as a blue white color. So now, okay, so they cannot be hydrolyzed, so they remain white color. Okay, all right. Okay, okay. So that is the end for the chapter 8. Okay, so that one is the DNA recombinant. All right. So this one no problem. This one just for your information. I will share this slide with you in our Telegram. All right. So for the reproduction, okay, you can expect there is we can get the picture. Maybe they give you a picture of flowers, okay, the sperms, okay, the secondary oocyte, okay, and that one is all is quite okay with you. Okay, I'm just worried if the question asks about the graph. Okay, the graph. This is the example of the graph. Okay, so show the graph of the concentration of hormone during the woman's menstrual cycle. Okay, all right. So it's a menstrual cycle. So they will have a follicular, ovulation, and luteal phase. So what you need to know first, identify what is the each graph represent. Okay, you are very lucky if the question give you already what each graph is represent. Okay, but I'm not sure about that. All right, so what you have to do now, just identify. Okay, so I'm confused. So just look at the highest one during ovulation. So of course this one will be the LH. Just identify. You no need according to the, oh, I must see the first one. Then I go to the second one. No need, no need. You just go which one, yeah. It's like a key point. Okay, so the search of the LH will cause the ovulation to occur. So this one will be LH. So this one will be automatically FSH. See? So this one will be my estrogen. Why this one my estrogen? Because whenever there is a growing of the follicle, so estrogen will be higher. Okay, so definitely this one will be the progesterone. See, simple. That. Okay, so don't go to the question unless you didn't identify all the graphs. Okay, describe the role of the hypothalamus into controlling of the menstrual. Okay, so hypothalamus, if you go back, they must start with the stimulate hypothalamus to secrete gonadotropin releasing hormone or GnRH. Okay, stimulate hypothalamus to secrete. Okay, why I have to start from hypothalamus? Because the question already mentioned here, hypothalamus. See, that's why you have to highlight. My story must start with hypothalamus. Okay, because some students they will say GnRH stimulate. Then maybe. Okay, maybe you start your point from GnRH will stimulate the anterior pituitary gland to secrete LH and FSH. Okay, yes, that one you can get the second point, but you missed the first point already. First point is who will stimulate the GnRH, because this question is asked you start from the hypothalamus. All right, so this is the second point as I mentioned to you just now. Okay, so because given mark is 2. That's why if you just straight away jump here, yes, you still can get the marks, no problem, but you already missed 1 mark. So sayang lah, okay? So you have to tamak. You have to grab as much as marks that you have. Okay? Alright, so now we go to the second one. With the reference of the hormonal pattern, okay, describe the example. Okay, so the keyword here, negative feedback, and involving the 2 hormone ovarian cycle. Okay, so as you know, the 2 hormone in ovarian cycle is progesterone and estrogen. Just something else. Okay, and I only want to focus about the negative feedback. So I no need to mention about the growing follicle release the estrogen. That one all no need. Okay, so what is the negative feedback here? The keyword is here. Increase. And see, ovarian cycle, so 2 hormone, that is another keyword. Let's say if here you only put only 1, okay, you forget to write about your progesterone, so actually you lost your mark. Okay. Then act on the both anterior pituitary and hypothalamus to inhibit. Okay, so to inhibit the FSH and LH secretion. So 2 marks. Okay. All right. Next question. Okay, so this one is also quite new for you in syllabus. Fertilization process in human. Okay, so during your SPM, I'm not sure you're discussing about this or not. But the menstrual cycle, ovarian, oogenesis, spermatogenesis, the plant, everything you already discussed. Okay, but for fertilization, the stages of fertilization, okay, you only discuss during matric level. That's why I say it's new for you. Okay, it's new for you. Okay. Before that, just I have to look. Okay, so now one head is entered already. Okay, so here is the formation of a polar body. Okay, and this one is the number of the sperm. Okay, so all the parts is here. Okay, you need to label all. Why? Okay, you are going to use to answer your question. Okay, so see here. Briefly explain the event during fertilization as shown in figure 9. Okay, so during fertilization. Okay, as you know, fertilization altogether we have 4 stages. Am I right? Okay, still remember? The first one, capacitation. The second one, acrosomal reaction. The fusion. Sorry, my head. Okay, so the biology teachers and handwriting always like this. Fusion of sperm head, okay, with secondary oocyte. Okay, so this one is my short form. Please don't do in your exam. All right, and last one, cortical reaction. Okay, you do like this. You're planning. Okay, I know the fertilization got 4 stage, but the question say in figure 4. Sorry, figure 9. Where is the 4? Figure 9, question 4. Okay, so here I already see this. Look at here. Okay, so the fusion of sperm head already entered the secondary oocyte. So I cannot write the capacitation. Okay, alright. Then acrosomal reaction. Oh, they dah pass dah. Okay, so they already passed the first barrier, so no problem. See, now I have to start focus from point 3 and 4. That's the important thing. Okay, so sayang lah if you write very perfect idea but start from the capacitation, actually you're wasting time answering the question, but you're not answering the question, you are answering what you know. Okay, so okay, so cortical granule release the enzyme. See, it start from here. Harden the zona pellucida. So prevent the polyspermy. Okay, so actually this one is the cortical reaction. Okay, alright. Then sperm head enter the secondary oocyte. So secondary oocyte continue meiosis 2 to form ovum. Why? Because here I already can see there is a formation of second polar body. Okay, so 1, 2, 3, 4, 5. See, actually you can score up to 5 marks here, but they always give you the maximum one. Okay, see, the points start from which, because you have to clearly understand the question asking what. Fertilization, but in figure 9, but the figure 9 is focused start from stage 3 and 4 only. Okay. Then the next question. Alright, so why B and C cannot enter? Okay, A dah berjaya. Alright, so fusion of the sperm C with the secondary oocyte trigger the release of calcium ion, which stimulate the cortical reaction. Okay, so cortical granule of secondary oocyte release the enzyme to the outside of the cell by exocytosis. So the enzyme harden the zona pellucida, which then function as the slow block. Okay, so prevent the polyspermy is the third polyspermy. Right? So this is the choice of your answer. Okay, so they give you 2 marks. Okay, but always remember, even though they give you 2 marks, but normally we have the planning. Okay, if student answer like this, okay, you can get marks. So, but make sure you still answer the question. Okay, answer the question. All right, and this one goes for the last question, I think. Okay, so briefly explain the level of hormone. All right, why I'm highlighting the hormone more? Because you have to really understand about the graph. Okay, about the graphs. So the diagram, I'm pretty sure you are quite okay because you can imagine the process. But once you get the graph like this, okay, you easily can get confused. Okay, so briefly explain, the level of hormone B remains high and hormone C drops at the end of pregnancy. Okay, so first, just answer first. This one is what? HCG, human chorionic gonadotropin. Okay, now this one, normally always at the high level will be the estrogen and low will be the progesterone. Okay, why you have to jot down? Because during your answer you didn't get confused. Suddenly you are very excited answer your question. Suddenly you put the C as your estrogen. Okay, so please be careful. Okay, so high level of hormone B stimulate the formation of oxytocin receptor on uterus. Okay, go back to the question. Hormone B remain high and hormone C drop. So ini high, this one drop. You just do like that. Okay, so you have to start high level of hormone B will causing the formation of a oxytocin receptor. Okay, then the decrease. Now you're answering. See, this is the first one here. Okay, so you're answering. Okay, here, first one. All right. So the decrease in progesterone receptor on uterus cause the decrease in hormone C. Okay. All right. So this is your progesterone. Okay. So now decrease. Okay. Progesterone receptor. Yeah. Then oxytocin will stimulate the parturition. Okay. All right. So 2 marks here given for your answering. Are you mentioned about the high of the hormone B? Yes, this is the high level of hormone B. 1 mark. Then, are you mentioned about the drop in the hormone C? Yes, drop in hormone C. 2 marks. Okay. What will happen if the production of hormone A ceases during the 4th week of the pregnancy? Okay, so hormone A, as you know, this one is the HCG. This one is controlled. Okay, this is the 4th week. Okay, this one 9 month. This one's a 9 month. Okay, so this one is the month. So if 4th week is middle here. Okay, so in this level, my HCG should be in a very high. Why? Why? Because what they can do? Okay, so just put us here. All right, so the level of progesterone and estrogen drop. Okay, why they can drop? Because corpus luteum will be degenerate. It's showing when the corpus luteum is degenerate, showing the HCG will be declined. This is okay. Automatically we know. Okay, this is still 4th week. See, 4th week, not 4th month. 4th week. Okay, so the first month of the pregnancy here is written here. Okay, so the level of progesterone and estrogen drop sharply, so miscarriage can occur. Okay, so this one we call as an analog question. Okay, analog question. So 2 marks is given. Right? All right. So I think it's already 9:28. Okay, so that's all for my discussion. Okay. So actually, I'm focusing how to answer the question. Okay, so if the question is more than 4 marks, it's considered as your short essay. Okay, if it's less than 3, it's considered as a structure. Considered as a structure. And you can expect structure means, okay, there is a lot of the short answer. Okay, just 1 mark. Okay, just explain everything. And please, whenever you are answering, please write in the full sentence. Full sentence. Okay, don't just write like a very short. Okay, okay, just have a look this one. Just for example, yeah. Explain the process of cloning in human gene. Isolate gene. Isolate plasmid. Don't do like that. Don't do like that. Okay, you have to put a very clear sentence. Okay, like I'm showing you the example like this. See, you give the full sentence. All right. And some of you can ask, Madam, can I write in the point form? Okay, so you can do like this. Okay, so you can do like this. Alright, so for me, I like to put the okay. I like to put the dot, showing it's the end of first point. It's the end of the second point. Okay, alright. So please focus the marks. Okay, and please write in the full sentence. Okay, don't do the ayat tergantung like that. Okay, high level of hormone B. Then after that is what? Don't expect you can get 1 mark. That no. Okay, so please remember when you are answering the biology, for each answer there will be a reason. Okay, you can ask yourself, am I giving the answer? Is it I can give the reason why I'm giving this one? So you should be okay. Okay, so please don't scared to answer the biology. Yes, it's a lot of theory parts, but actually it's very easy because the biology means life, this is your life. Okay, whatever happened in our side, okay, they are asking in form of question. All right. So I think that's all from me for today. Okay.
Okay, so sorry Okay, terima kasih kepada Madam Nashimah atas perkongsian beliau yang banyak memberi tip-tip yang amat berguna dalam menjawab soalan biologi semasa PSPM nanti. Okay, so pelajar-pelajar di ruangan YouTube, okay, adakah anda rasa Madam Nashimah ini hebat? Kalau rasa ya, boleh tak you tuliskan 1. Kita bilang ada berapa 1 kat situ. Okay, adakah kamu merasa perkongsian beliau membantu anda lebih memahami macam mana nak menjawab tepat kepada kehendak soalan So saya rasa ini adalah perkara yang penting Betul tak Madam Nashimah? Betul Okay. Okay, so sekarang kita pergi ke sesi soal jawab. Okay, saya ada beberapa soalan yang student tanya. Okay, so tanpa melengahkan masa, kita pergi ke soalan yang pertama. Okay, dari Kai Kim.
Okay. So if produce same sticky end which is GAATTC and CTTAAG. Madam, if we write. Okay, means are you asking about the question if why we are using the same restriction enzyme? Am I right? Okay, so if they ask, okay, please don't write GAATTC and CTTAAG. No, don't do like that. Okay, because when we mention at the base for the palindromic sequence, the base we only refer GAATTC. Okay, so CTTAAG normally be silent because it's the second strand. Okay, we only highlight the base. Okay. So just write GAATTC. Okay. So if you want to write produce same sticky end, better you just write to produce complement sticky end. Complement sticky end. Just leave it at that. The terms.
Okay. Okay. Thank you very much. Okay. The second question is from Xiao Yang Chang.
Can we left the drawing of recombinant DNA with both open ends? DNA recombinant. Can we left the drawing? What do you mean by this? Drawing the recombinant DNA with both open ends. Are you means you are drawing, isn't it? Oh, okay. So better you do in the fragment form lah. But once you already put like a box, okay, so we understand that it's a fragment. Okay, so it's better for you to just close your end. Okay, so you see the fragment clearly. Okay, so if like see in our college they are doing some project. Okay, so they will do how the end is look like. So see, am I have the example here? Okay. So for like example like this, see, all right. So this is how the ChemS doing the fragment. Okay, so better you just cut or you close the both fragment. Okay, easy for student to understand. Okay.
Okay. Okay, thank you. Okay, the third questions.
We forget the full name. Can we directly write? Okay, yes.
Mention
But please make sure G and RH. G capital letter, N must be small letter, then RH capital letter. Yes.
Okay. So then we proceed with the questions from Chi Chun.
When we mention the name of enzyme, we have to state in the full name or short form is acceptable. Okay, for like just now question. So like a gonadotropin releasing hormone. So you can write as a GnRH. But better it's good for you if you write as a gonadotropin releasing hormone in bracket GnRH. Okay, then after that, the next sentence you just can simply use the short form. Okay. But let's say you already forget, so don't forget. Try to write full name. But if cannot, last choice, don't leave it blank. So you just write the short form. But make sure the all the capital letter, small letter, all you follow.
Oh, cannot change lah. Okay. Can I use trigger instead of
This one just a normal can. But normally,
can?
for example, let's say we are using the enzyme. Okay, so enzyme we always use to catalyze. That is like standing a word. Okay, so enzyme will catalyze. Trigger, stimulate. Okay, can. All right, so we normally use that types of terms. HCG is only in the first trimester. Yes, first
Madam, if
trimester.
we write
But you
like
have to understand the first trimester is how long? First trimester is uh, the graph just now is showing the month. Okay, yes, HCG only the first trimester. Then after that, the HCG, the corpus luteum will degenerate and placenta will take the function. So there is no more HCG.
okay.
To produce palindromic base sequence for the reason to use same restriction enzyme for plasmid and target gene. Palindromic base sequence. What if we write to produce palindromic base sequence for the restriction enzyme? To produce the palindromic, no. Your answer is like a tergantung little bit. To produce palindromic base sequence. Okay? So actually, why you are using the same restriction enzyme? Actually, to get the both end, which is complementary. So once you mix, okay, complementary or compatible. Okay, so maybe this one you're using the eco R1, you get the sticky end. This one you're using the small one. Okay, so to produce the palindromic base sequence. So maybe here is CCC this is the small one. Here is the eco R1, you get the sticky end GAATTC. Okay, so cannot. Better you stick for that answer.
Okay. So the next question is, I think ini Nurul
How
Suhada, the
many?
comment essay.
Okay. Actually,
Comment.
we are in the new syllabus. Okay, so new syllabus. We are not calling as an essay lah. We just call it as a short essay. Okay, you can refer back to my clone question that I shared to you in our Telegram. Okay, if you look at the first page, I already give you the allocation. The first question, how many marks? Okay, so normally for question that we discussed today, okay, so we have a 60, 40 marks. So that is a possibility to get the essay. Essay means here is only up to 4, 5 marks only. Okay, it's like that. So you just
Short
expect there only will be 9 questions. 9 questions. So we just omit the chapter 6 only. Okay, so chapter 1 until chapter 10 will be there, 9 questions. So then you just have to understand that they will be beranak pinak lah. You just ready for that. Okay, so we cannot expect. Sometimes there is no essay at all. All is a short answer. Okay, and for example, inheritance, you maybe can expect because normally for one inheritance, they give you up to 4, 5 marks. So that way is considered essay, short essay lah. We call it as a short essay.
Short essay lah. Okay. So we have the 2 more second last questions. Okay, yang ini Muhammad
The
Firdaus.
question needed to give
Need the
2
candidate
roles.
to give 2 roles. Oh.
Then the candidate give 3 answer role and 1 answer. This one yang I highlight just now. Okay, thank you Muhammad Firdaus to asking that. Okay, so please, if you get like this 2, but you give 3, unfortunately, the one of the answer is incorrect, so they actually will cut. That one we call as a wrong answer, right? Okay, they will cut the mark. So sometimes you possible to get 0 mark. Please be careful about that one. So if you get the open question like just now I'm sharing to you, it's very easy. Okay. So please be careful if they give you 2, if you want to give 3, make sure you really confident with your answer. Okay. And not like SPM, yeah, I know. Previously in SPM they can. Okay. So in matric, no.
Oh, cannot. So this one have to take notes, all the candidates. Okay, the last question is from Sandra.
The polymerase added to synthesize the second strand to complementary, or is that reverse transcriptase enzyme? Okay, reverse transcriptase we add from mRNA. From mRNA to produce the DNA. That is a reverse transcriptase. That's why I'm giving to you in my answer just now. I just give you the reverse transcriptase. Okay. Actually, to identify, are you alert or not? Okay, good, you are alert. Actually, that is the DNA polymerase. Okay.
Okay, so I think there is no more questions from candidates. So majority mengucapkan membantu. Thank you Madam Nashimah. Thank you and so on. So I think sebelum mengakhiri sesi kita pada malam ini, so kita akan nampak banyak thank you. Okay, thank you. Okay, so boleh tak? So kalau you rasa Madam Nashimah ini betul-betul membantu anda, boleh tak? Sebab kepada kita nampak sini ada 239 pelajar kat sini Boleh tak one person give her a like? Okay, a like satu like bagi dia. Okay, simple aja, you just click. So satu like bagi daripada kamu actually memberi banyak motivasi kepada Madam Nashimah sebab okay, so your like actually is important. Boleh tak? So every one of you ambil tindakan and give her a like. Okay, to show that you already appreciate Okay, so before we end the slot, okay, so remember to give her a like and bagi sesiapa yang belum subscribe channel kita, bolehlah subscribe channel kita untuk mengikuti kita punya slot yang seterusnya. Okay, dan jangan lupa mengisi borang penilaian untuk slot hari ini. Okay, hari ini pilih tarikh hari ini dan speaker adalah Madam Nashimah So jangan pilih orang lain pula. Okay, so sila isi borang penilaian. Okay, your penilaian actually memberi kita banyak manfaat untuk kita membuat penambahbaikan untuk slot yang akan datang. Okay, so terima kasih atas sokongan anda selama ini. So wish you good luck in your PSPM. And before end, I want to show you a slide Okay. Yang ini. Mana dia? Okay. Your future is created by what you do, not tomorrow. So I like this phrase very much. Okay, saya rasa you punya tindakan akan mempengaruhi you punya kejayaan pada masa yang akan datang. So harap pelajar-pelajar mulai hari ini. So you ada 7 hari lagi. So if you want to make a difference, you can make a difference. So mula dari sekarang, saat ini untuk buat persediaan yang rapi untuk menghadapi peperiksaan anda. So we all here wish you good luck and 4 flag is for you. Okay, if you take the action today bukan tomorrow, is today Okay, good night everyone and good luck.
Okay, good luck everyone.
Okay, good luck. Okay, bye.
Bye.
Okay.